Thanks for having a chat with me today on this beautiful day at UC Davis. Remember, most of the world has a perception that research scientists are completely reactive. They keep digging until they find something interesting, but I think we both know that there's a lot of strategy built into research. Can you talk to me a little bit about the strategy that you employ with your research program?
Yes, I will talk to you about that. As a researcher myself, we do not talk about research as just we go into the lab and we start just any random thing and see what comes out of it and then go on from there. We have a plan about what we want to do. Every researcher has a research project that they work on, and in the team, there is a team, or in the lab, individuals contribute to that team project. And in my team, which is the Walling Lab at UC Riverside, we work with a protein called leucine aminopeptidase that's induced in tomato leaves after mechanical wounding and caterpillars feed on it. Anytime there is any mechanical stress on the leaf, the protein is induced. We call this leucine aminopeptidase, or LAP-A for short. Now, there are many roles that play into studying this protein, and I take a piece of that puzzle, and there are other graduate students that do the same. 'Cause I'm a graduate student, and there is another project in the Walling Lab that's also involved in whitefly and plant interactions. So now I want to go back to the LAP-A project. I want to learn how this protein is affecting the plant immune responses. So what other proteins it is interacting with. And I do various studies at the same time, protein-protein interaction studies, affinity chromatography, co-IP, and from those we find that there are many candidates that we are working on. And then from those candidates, we are honing down on which ones are the most important that play a role in the model that we have with LAP-A possibly interacting with another protein. So we have thousands of proteins that we think might interact. But then from those, we are honing down, and we want to look at the big picture, which is how is LAP-A playing a role in the plant immune response in tomatoes? But for that to be answered, we need to have several people pitch in. We all have different projects that all align with the big picture. And that's the only way we can answer the question about how LAP-A plays a role in plant immune response in tomatoes.
A true team.
Yes.
A true team. You mentioned you work with Dr. Walling, whom I know you consider a mentor.
Yes.
Why is it important for students to have a strong mentor, and how did you choose yours?
I think it is very important for students to choose a mentor that fits their needs, because everyone has different personalities and different skills. So it is always good to be with someone, an advisor, that can fulfill those needs that the student has. And every student has different, both things that are good and bad in terms of, for example, there are some students that might need more guidance in terms of writing, scientific writing. And if the professor is willing to put in that time to help the student with the writing skills, then that is a good fit. And there might be others that are good in writing, but they don't really need so much guidance.
Right.
And those professors are not so much there, but they help them in other ways. And then, but I think that both those, in both scenarios, they're good mentors.
Sure.
It just depends on the person and what the needs are. Now, in my case, I consider Linda to be a mentor because I met her at a class at UCLA. I was an undergraduate at UCLA, and she was an invited speaker in this plant microbial interactions class that I was taking with Dr. Ann Hirsch, and Dr. Ann Hirsch invited Dr. Linda Walling, and I was actually a junior in my undergraduate career at UCLA. I listened to her talk and I thought it was amazing that she was talking about plant and insect interactions. I never thought that I would do this in the future, but it just happened to be that later on I started to really consider doing plant biology research. As an undergraduate, I was in Dr. Ann Hirsch's lab, and then I started to look around and I saw that UC Riverside had a great plant biology research program, just as UC Davis does. both UCs have very strong agricultural. Um, we can say they're agricultural campuses, both UC Davis and UC Riverside. So I decided to apply to UC Riverside, and I considered applying to her lab. I reached out to her. I told her that I was interested, that I had listened to her talk like a year ago, and 1 or 2 years ago, I don't exactly remember when that was. I sent her my CV, and then we did an interview, and I joined her lab
Awesome.
as a graduate student researcher.
Great
Yeah.
story. So tomato is a key focus for you in your research.
Yes.
Can you talk a little bit about the changes that have been brought about by having a mapped tomato genome since 2012? How does that affect your research?
It affects my research a lot because, as I said, I'm working with this protein called LAP, and we want to know what other proteins it's interacting with. And one of the studies that I did was to find out what proteins it's interacting with by immobilizing this protein on beads and then pouring leaf protein extract onto that immobilized protein and find what it's interacting with using this high-tech machine. And many proteins were discovered, and the only way we could have discovered that those were possible interactors was due to having the proteins annotated in Soul Genomics. Otherwise, we wouldn't be able to do that. We wouldn't be able to have these possible interactors for LAP-A.
Right, excellent. Speaking of Soul Genomics, we are at the Soul Genomics conference here at UC Davis, 13th annual. Two-part question for you. One, why is it important to come together with like-minded colleagues at an event like Soul Genomics? And if you just pick off one thing, what would be the number one takeaway that you have from your time at the conference here at UC Davis?
First of all, I am very grateful to come here to Soul Genomics, to have had. I was invited by the Soul Genomics. I'm not sure how to. The staff that organized this conference.
Organizing committee, yep.
Yes, the organizing committee. They sent an email to my PI saying they were looking for a co-chair for us to co-chair a session along with another chair, and they were willing to let me participate in this. Now, I was very enthusiastic about it because Soul Genomics has helped my research a lot, a tremendous amount, like I just explained in my previous answer, and I am grateful that I am here meeting like-minded people because I think that is the only way that we can put the face to the name of those papers that we read. And when I have a question about something that comes up when I read an article and I don't know about the methods, I can send an email and get a response, but it's not the same as interacting with the authors of those papers that I read and networking with them and just talking about my project and them sharing their thoughts about how I should go, what is the best path to follow in my project, or I can ask them questions about their papers. And it's just great to have this big community that gets together once a year in Soul Genomics or in other scientific conferences as well, to just talk about science and to see that there are many things that we all share the same and that we can contribute to one another. We can help each other's projects out that way. And I think the number one thing to take from this is networking. I learned that early in my undergraduate career. The first conference that I went to was botany, which was for the BSA meeting. And I learned that it's great to interact with other people and get to know other people, know them and keep in touch with them, because we can help each other's projects, and in the long run we're going to help each other answer those questions that are those big picture questions. Like in my project is, how is LAP-A important in tomatoes? And in other projects that there are other questions, and I might not have the same project as other people, but we all want to answer those research questions and we can all help each other out.
Absolutely. Well said. Thanks very much for having a chat today, Irma. I appreciate it.
You're welcome. Thank you.